We're going to continue our discussion of genetics, and we'll also start to discuss several inherited diseases. At the end of this lecture, you'll be able to define numerous terms that are listed on this slide. Also, you should be able to list Mendel's laws of inheritance, interpret a simple pedigree and determine the inheritance pattern based on a pedigree, and also draw a Punnett square and determine genetic ratios. This slide shows the general terminology that you will see in a pedigree. Now, there are several principles of inheritance. So basically, from each chromosome pair, one chromosome is inherited from each parent. So you get one maternal and one paternal chromosome per set. Each individual has 2 copies of each chromosome, so you have 2 copies of each gene. Meiosis reduces cells to only 1 copy of each chromosome. There's a 50-50 chance that each chromosome will be passed to the next generation. Some definitions are as follows. Congenital diseases are diseases with genetic components that are present at birth. However, keep in mind that a genetic disease may not be a heritable disease. An example of a congenital disease that's not inherited is spina bifida. A carrier is a heterozygous individual who has no apparent abnormality, but they can pass on an allele for a recessively inherited genetic disorder. Penetrance is the proportion of individuals with a gene mutation who actually express the disease condition. Founder effect is a high frequency of a gene mutation in a population founded by a small ancestral group when one of the founders was a carrier of a mutation. Incomplete dominance is when offspring show traits intermediate between the 2 parental phenotypes. One allele of a heterozygous pair only partially dominates the expression of its partner. So for example, you have a red flower and a white flower. So instead of producing either red flowers and white flowers, you can produce pink flowers. So that's an example of incomplete dominance. Codominance is a pattern of inheritance where both alleles are expressed. So both alleles will produce an effective product. So for example, a person that has blood type AB has both A and B antigens on their red blood cells. The P generation in a pedigree is the parental generation. The F1, the first filial generation, is the first generation of offspring. The F2 is the second, and you could keep going, F3, F4, depending on how large that pedigree is. We already went through the laws of inheritance, but let's get a little bit more specific with Mendelian inheritance. So in Mendelian inheritance, each individual has 2 factors for each trait. We already said that, one from each parent. The 2 factors may or may not contain identical information. If the 2 factors are the same, the individual is called homozygous for that trait. If the 2 factors have different information, the individual is considered heterozygous. Alternative forms of a factor are referred to as alleles. The genotype of an individual is made up of many alleles that it possesses, and an individual's physical appearance, what's called the phenotype, is determined by its alleles as well as the environment. To continue with Mendelian inheritance, an individual possesses 2 alleles for each trait. One allele is given by the female parent and the other by the male parent. When the gametes form, the paired alleles separate randomly. So each gamete receives a copy of one of the two alleles. The presence of an allele doesn't guarantee that the trait will be expressed in the individual that possesses it. In heterozygous individuals, the only allele that's expressed is going to be the dominant allele. The recessive allele is present, but its expression is hidden. There are two laws that make up Mendel's laws. The first law is the law of segregation. And in the law of segregation, when any individual produces gametes, the copies of a gene separate so that each gamete receives only one copy. A gamete will receive one allele or the other allele. The second law is the law of independent assortment. And in the law of independent assortment, alleles of different genes assort independently of one another during gamete formation. There are various types of genetic disorders. Single gene disorders cause things like biochemical defects and metabolic disorders. Polygenic disorders are usually congenital malformations or diseases that are caused by numerous gene disorders, such as diabetes and cancer. Chromosomal disorders usually lead to birth defects. For your chromosomal disorders, that is usually the result of a loss, a gain, or abnormal arrangement of one or more chromosomes. And these chromosomal disorders can cause excessive or deficient amounts of genetic material. We discussed chromosomal structural alterations in the last lecture, but as a review, you could have inversion, where a chromosomal segment separates and is reinserted at the same place but in the reverse direction. Duplication is when you get doubling of a chromosomal segment. Deletion is breakage or loss of a portion of a chromosome. In translocation, you have a breakage of two chromosomes with transfer of the broken parts to the opposite chromosome. And you have isochromosome, where you have a splitting at the centromere during mitosis, so that one arm is lost and the other is duplicated from one chromosome. You also have changes in chromosomal number. So in this you have aneuploidy, which is excess or loss of one or more chromosomes. You have monosomy, where you have only one of a particular type of chromosome. So an example of monosomy is Turner syndrome, where an individual would only have a single X chromosome. There's also trisomy, where you have three of a particular type of chromosome. And an example of trisomy is Down syndrome, where you have three of chromosome 21. In non-disjunction, you have your failure of chromosomes to separate, and there's also polyploidy, where you would have more than two complete sets of a chromosome. Other chromosomal abnormalities include mosaicism and chimerism. Mosaicism is two or more genetically distinct population of cells from one zygote in an individual. This is the result of early segregation errors during fertilized egg division and is more common with sex chromosomes. Chimerism is two or more genetically distinct population of cells from more than one zygote in an individual. In autosomal dominant disorders, the dominant gene is located on an autosome. Affected individuals have an affected parent. Each child of an affected parent has a 50% chance of inheriting the abnormal allele. Unaffected individuals don't have affected children. Males and females are both affected equally, and the inheritance pattern is vertical. In other words, every generation has affected individuals. Some of the characteristics of autosomal dominant disorders are that the affected children will have an affected parent. Heterozygotes are affected. Two affected parents can produce an unaffected child, and two unaffected parents will not have an affected child. So here's a pedigree showing an autosomal dominant disorder, and you can see that there's disease, the colored in circles or squares, in every single generation of the pedigree. Here's another pedigree of an autosomal dominant disease, and you could see that affected offspring always, in this case, have affected parents. Each child has a 50% chance of being affected. If the parents are not affected, then the offspring usually are not affected. Males and females are both affected equally. So in this case, there are 4 males and also 4 females. And you could see that vertical line. In other words, every single generation, when you look up and down the pedigree, there are affected individuals. Some examples of autosomal dominant disorders are Huntington's disease, neurofibromatosis, meiotic dystrophy, familial hypercholesterolemia. There are numerous autosomal dominant diseases. We're going to briefly mention several genetic diseases. However, we're not going to discuss them in detail right now. We will just talk about a couple of the characteristics of certain diseases. We are going to talk about these diseases much more in depth later in the semester when we're talking about molecular testing of inherited disease. So the first disease we're going to mention is neurofibromatosis type 1. This disease affects 1 in 3,000 people, and people that have this disease have tan or what's called café au lait, skin spots at birth. Most cases have mild symptoms such as skeletal deformities, eye and ear tumors that might lead to blindness and hearing loss, and possible learning disabilities. This disease is in a gene on chromosome 17, and the gene is a tumor suppressor gene that controls cell division. In Huntington's disease, this affects 1 in 20,000 people, and this disease leads to progressive degeneration of brain cells. It causes severe muscle spasms and personality disorders, and it ultimately results in death within 10 to 15 years from the onset of symptoms. It's caused by a gene located on chromosome 4. And in this gene, you have numerous repeats or trinucleotide repeats. And these repeats are CAG. And the severity of the disease depends on the time of onset and the number of triplet repeats found within the gene. Now we're going to discuss autosomal recessive disorders. These disorders are when you have a recessive gene residing on an autosomal chromosome. So affected members of a family are usually siblings of the proband. Carrier parents have a 25% chance of having an affected child. Affected individuals may have unaffected parents. So in other words, in autosomal recessive disorders, the disease can skip generations. So that inheritance pattern is not vertical, it's horizontal. So the inheritance pattern affects many people in a generation, but then can skip a generation. So some general characteristics: most affected children have normal parents, since the heterozygotes have a normal phenotype. Two affected parents will always produce an affected child. So here is a typical pedigree of an autosomal recessive disorder. Here's another pedigree of an autosomal recessive disorder, and you can see here that carrier parents have a 25% chance of having an affected child. Males and females are affected equally, just like in autosomal dominant disorders. Affected individuals can have unaffected parents, and if both parents are affected, then all of the offspring are going to be affected, and this is a horizontal line. Some examples of autosomal recessive disorders are cystic fibrosis, Tay-Sachs disease, Gaucher's disease, hemochromatosis, sickle cell anemia, and phenylketonuria. Cystic fibrosis is a disease that causes severe lung damage and nutritional deficiencies. Approximately 30,000 adults and children in the United States have cystic fibrosis. The problem with cystic fibrosis is you get an accumulation of mucus in the lungs that interferes with gas and ion exchange. What it also does is it causes colonization of the lungs by pathogenic organisms such as Pseudomonas aeruginosa. Cystic fibrosis is caused by a mutated gene on chromosome 7. In the disease hemochromatosis, there's an overabsorption of iron from food, and this iron accumulates in the organs in the body and causes damage. So hemochromatosis is usually treated through phlebotomy. It's caused by a dysfunction in the hemochromatosis type 1 gene, which is either called HFE or HLA-H. The HFE codes for a membrane-bound protein that binds with beta-2 microglobulin and transferrin. So without proper HFE function, the cell is not going to sense that it has iron, and so you're going to absorb more iron. And phenylketonuria, this happens in 1 in every 5,000 births, and there's a lack of an enzyme that's needed to metabolize the amino acid phenylalanine. This leads to the accumulation of the amino acid phenylalanine in brain nerve cells. So it impairs nervous system development, and this is caused by a mutated gene on chromosome 12. Sickle cell anemia is an example of a disease caused by one gene and there's one mutation. So the sickle cell hemoglobin was discovered in 1949, and there are over 400 human hemoglobin variants. Sickle cell anemia is due to a single amino acid substitution in one of the globin chains. It causes increased mortality and morbidity in individuals in Africa. So in sickle cell anemia, you could have either sickle cell trait or sickle cell disease. In sickle cell trait, these individuals are heterozygous, they have a normal life expectancy, and may have some blood in the urine occasionally. In sickle cell disease, this is homozygous. There's lifelong hemolytic anemia, increased risk of infection, and individuals have a variable course. There are compound heterozygotes with both hemoglobin C and hemoglobin D. In sickle cell disease, you have the sickle cell hemoglobin, which differs from your normal hemoglobin by a substitution of valine for glutamic acid at position 6 of the beta globulin gene. So in the beta globulin gene, you have an A to T substitution, which causes valine to become a glutamic acid. and intracellular fibers cause the sickle cell deformity in the red blood cells. You can see here the normal sequence compared to the mutant sequence. So in the normal sequence, we have GAG, which codes for a glutamic acid. In the mutant sequence, that A is substituted to a T, and GTG is encoded as a valine. The next disorders we're going to discuss are X-linked recessive disorders. So in these disorders, the affected individuals are predominantly males. All female offspring of an affected male will carry the disease gene. So these are obligate carriers. The male offspring of affected males are always unaffected, and the inheritance pattern is neither vertical nor horizontal. This is an example of an X-linked recessive disorder, and you could see that females are carrying the disease and males are affected. So when you see a pedigree where it's almost all males, then you have to think X-linked recessive. In X-linked dominant disorders, all female offspring of affected males are going to be affected. No male offspring of affected males will be affected. An affected female has a 50% chance of having an affected child, and all affected individuals have an affected parent. And here's an example of an X-linked dominant disorder. So you can see in this disorder, unlike X-linked recessive where it's almost always males, in X-linked dominant you can see affected males and affected females. So all daughters of affected males are affected, but not sons. The affected female has a 50% chance of having an affected child. Unaffected individuals will have unaffected offspring, but affected individuals will have an affected parent. Some examples of X-linked disorders are color blindness, hemophilia, Duchenne muscular dystrophy, Fragile X syndrome, and numerous others. In Duchenne muscular dystrophy, this affects 1 out of every 3,600 male births. It's characterized by wasting of muscles that eventually leads to death. This disease is caused by a lack of dystrophin, which causes calcium ions to leak into the muscle cells. This gene is passed from a carrier mother to carrier daughters. In Fragile X syndrome, the X chromosome is nearly broken. This disorder is most common in males, and females that have the disorder have much less severe symptoms. The symptoms include learning disorders, a long face, large testes, and protruding ears. It's caused by one of these trinucleotide repeats, so it's an excessive number of repeats of CGG. There are up to 230 copies in the disease compared to in normal individuals who have 6 to 50 copies. There are also diseases that are caused by abnormal sex chromosome inheritance. And 2 examples of this are Turner syndrome and Klinefelter syndrome. So in Turner syndrome, females have only 1 sex chromosome. Individuals with Turner syndrome are usually shorter in stature, have a broad chest and a webbed neck, and also have non-functional ovaries. Klinefelter syndrome occurs in males who only have 1 Y chromosome and 2 or more X chromosomes. The disease causes sterility, large hands and feet, and long arms and legs. Diseases can also be inherited through the mitochondrion. These types of diseases are inherited only from the mother, so all offspring of an affected mother are going to be affected. So here's an example of a mitochondrial associated disorder. Examples of mitochondrial associated disorder are Leber's hereditary optic neuropathy, Cairns-Sayre syndrome, mitochondrial encephalopathy, lactic acidosis, and stroke-like episodes, and myoclonus epilepsy. So you can do mathematical calculations or probability to calculate a genetic risk. So the probability, or P, of two independent events, A and B, occurring at the same time can be calculated. Or the probability of either of two independent events. And those are the two mathematical formulas. You will not have to calculate genetic risks or probabilities for this course. You will, however, calculate genetic risks using Punnett squares. So here's an example of a Punnett square where you have two carrier parents of an autosomal recessive disease. So this is going to be big R little r crossed with big R little r. You can fill out your Punnett square and determine that 1/4 of the children would be normal, 1/4 or 25% have a chance to be affected, and 50% will end up being carriers. The genetic risk of having one normal parent and one carrier parent of an autosomal recessive disorder would be big R crossed by big R little r. And in this situation, you would have a 50% chance of having a normal child and a 50% chance of having carriers, and none of the children will actually be affected. The genetic risk of having two heterozygous parents of an autosomal dominant disease would be big D little d crossed with big D little d. When you fill out your Punnett square, you would find that there's a 25% chance of offspring being normal, and that's the little d, and a 75% chance of offspring being affected, and that is either the heterozygous big D little d or homozygous big D big D. In an autosomal dominant disorder, when you have one heterozygous parent and one unaffected parent, that would be big D crossed with little d, you would have a 50% chance of having a normal child and a 50% chance of having an affected child, and there would be no carriers because autosomal dominant diseases do not have carriers. So we talked about Mendelian laws of inheritance, and many diseases are inherited in a Mendelian fashion. However, there are diseases that are inherited in a non-Mendelian fashion. So in these diseases, there are genes that are unstable, and they can change from one generation to the next generation, so you can't really predict what they're going to do. This would be considered premutation. And some diseases that follow this pattern would be Fragile X syndrome and other trinucleotide repeat disorders. Also in non-Mendelian inheritance, the law of independent assortment is violated by genes that are closely linked or very close together on the same chromosome. Also in non-Mendelian inheritance, two members of a single gene pair can be found in the same gamete, and that's called uniparental disomy. And some disorders that follow this non-Mendelian pattern would be imprinting, which is differential expression of genetic material depending on whether it's maternal or paternally derived. Engelmann syndrome, which is a deletion or a mutation of maternal chromosome 15 and a failure to express maternal chromosome 15. And Prader-Willi syndrome, which is a deletion or a mutation of paternal chromosome 15 and a failure to express the paternal chromosome 15. So we talked a little bit about the probabilities, and you can do a lot of calculations using Hardy-Weinberg law, where you have your P is the frequency of having a normal allele, Q is the frequency of having a diseased allele, and you can do numerous calculations based on the P's and the Q's. However, we will not be doing this type of calculation. We'll only be doing our genetic ratios based off of a Punnett square. If you were to do the Hardy-Weinberg calculations, you can have a disease such as this one. And if you wanted to determine the chance that a specific individual is going to carry the mutation, you can follow the mathematical calculations and determine that 1 out of every 632 individuals are at risk of carrying this mutation. Here's an example of a pedigree. We've seen numerous pedigrees in this lecture. In this pedigree, the disease is X-linked. So what you should be able to determine for a quiz or an exam when you're given a pedigree, is it a dominant disease, a recessive disease, who might be carriers, who definitely have to be carriers, who's affected. So these are the types of questions you should be able to answer. The prevalence of common genetic diseases are listed on this slide. And you can see that some genetic diseases are highly prevalent in the population. And we are going to discuss much more specific details of several of these inherited diseases later in the semester when we talk about molecular detection of inherited disorders.